Question 1, model answer. We start off with just some definitions. Lots of nearly right definitions. Lots didn't mention the word evolved, so we'll try and incorporate that into the answer. So let's start off with homologous. So these are structures evolved from the same original structure. Structures evolved from the same structure but may have different functions. So those are like your arms and the wings of a bat. Same pentadactyl limb but different functions. And analogous, so they are evolved from different structures, but they're evolved to carry out the same function. So evolved from different structures but same function. So those are like the wings of a butterfly and the wings of a bat. They both fly, they're both wings, but they're completely different structures. You didn't need these examples in there, and indeed there isn't any space. So if you're writing the answers down the side, you've done enough for the 2 marks there. Lots of problems putting the kingdoms in. Well, let's start with what we hope are the easiest. So we're not going to put prokaryotes in here. It's telling us in the question that the bacteria and archaea were grouped into the prokaryote, whilst the eukaryotes were formed into 4 different kingdoms. So the answers are not prokaryotes, it's the other ones. So there's quite a few answers with prokaryotes in. You need to use this information. What's got a cellulose cell wall with chloroplasts in and makes its own food? And lots of different tissues, so multicellular. Well, that's got to be plants. So plantae. If you just write plants, it's fine as well. And then things without cell walls that eat other things, heterotrophic. Well, that's going to be our animals. Animalia. Then these are the ones that prevented a little bit of a problem. Well, chitin or chitin, that should have just given it away straight away. That's our fungi. So fungi don't have chloroplasts and live on the food. They digest other organic compounds. They're heterotrophic. So that's fungi. And then this is the one, the leftover one, but doesn't show any tissue differentiation, so mostly unicellular. Cellulose cell wall, if it's there. Some are autotrophic, so that's the odd group, the Protoctista. And if you're getting those wrong, that's your AO1 revision that needs looking at. More AO1 stuff on the right hand side. The name for that type of diagram, phylogenetic or phylogenetic, or sometimes even called a cladogram, but phylogenetic or phylogenetic tree. This theory here is not the 5 kingdom theory. The bacteria, the archaea and the eukaryota. So that is 3 domain theory. You needed more than just the domain theory, the fact that you are putting them into 3. And the classification, as it says, is largely based on the gene coding for a ribosomal subunit. So gene coding, this ribosome is made out of nucleic acid, and the analysis of the gene, or the gene is the DNA. So we're looking at DNA, comparing DNA. What do they look for when they compare DNA? Well, they are comparing nucleotide base sequences of the DNA in the gene. So anyone who said amino acids or anything like that, is way off. So this is DNA. So compare nucleotide base sequences in the gene. You could have got away with just base sequences, or comparing the base sequences. And then we've got a second mark. Well, how is it support? How are we putting things into our domains? So they're looking for organisms that have similarities will be put into the same domain, or the other way around. Differences will be put into different domains. But you need to say that's the evidence for this modern theory. So organisms within a domain, so within the domain of bacteria, have greater similarity to each other. So quite often the first mark was got there, but the second mark with difficulty explain that. But if you do lots of past questions in the Moodle, that comes up quite a few times. Then we've got a question where a lot of students got 2 marks out of these 3. The first mark is what I'm sure you've put already, is that for the archaea you've correctly guessed that it's got the ether bond and of the normal or branched. You're going for branched. So the first mark is just coming up with, I think that, so the phospholipids would have ether bonds as opposed to the ester bonds, and branched fatty acid tails. So that's getting you the first mark. Now for the other 2, it is quite straightforward. If you need to use this information, all the relevant bits of information here. So we need to talk about the ether bond. So let's talk about this ether bond. And I highlighted it in the Help Me video. So it's resistant to hydrolysis, so it's resistant to sort of chemical digestion or breaking down, and it's also less reactive at high temperatures, so it's going to be more resistant, it can live at higher temperatures before it breaks down. So that's what we're going to say. So more resistant to chemical effects, e.g. hydrolysis and high temperatures. And that ties in with our information. We're living in hot springs that might have a high or low pH. And let's talking about, this is the rarer mark to get, the branched phospholipid. So the branched fatty acids. So branched fatty acids. Well, these molecules at higher temperatures are less able to move. So it says there they're less fluid. So less fluid, so less able to move. And you know that when things get hot, they move, and then you might have done this experiment in the lab, and that will then break down and make the membrane permeable and not good at doing its job. So less able to move, so will not break down as easily when hot. And so it won't be permeable, it will maintain its integrity. So not permeable. That's what it says in the mark scheme, but I think that description will do.